Hello everyone. Uh, as per our last lecture, we have started the chapter inflammation in pathophysiology. Right? In that, we have studied the definition of inflammation, the symptoms of inflammation, or you can say five signs of inflammation. There are two types of inflammation, acute and chronic, and we have started the acute inflammation. Acute inflammation occurs in two types of events, vascular events and cellular events. From that, we have completed vascular events. So, vascular events, I will summarize first. Vascular events include two types of changes, hemodynamic changes and the altered vascular permeability. The hemodynamic changes include five steps, means the changes occur in the blood flow and the diameter of blood vessels, right? And that occurs in five steps. And altered vascular permeability involves the five mechanisms which are responsible for the change in vascular permeability, right? So today we are again continue with the next topic, and that is cellular events responsible for acute inflammation. Cellular events, the name itself suggests, occurs when there are the changes related to the cells, and we have already discussed that the cells, particularly WBCs, and in that also neutrophils and lymphocytes are mainly responsible for the inflammation. So here, whenever there is any changes occur related to the leukocytes, or you can say WBCs, they are cellular events. So cellular events also includes two types of processes. First, exudation of leukocytes, and second, phagocytosis. Starting with the first one, exudation of leukocytes. The definition is the escape of leukocytes occur from the lumen microvasculature to the interstitial tissue is the important feature of inflammatory response. That means what happens? I think I have also explained the exudation and the transudation. What is the difference? Both are the movement of fluid from the blood vessels into the tissue. But what is the difference is transudation is the movement of fluid along with a very small amount of only plasma proteins. There are no cells present during the transfer. While the fluid transfer from blood vessel to the tissue along with cells, along with WBCs, that process is called as exudation. So here the exudation of leukocytes is the transfer of WBCs along with fluid into from the blood vessels into the interstitial tissue. That means the nearby tissue to the blood vessel. So when the WBC is transferred from blood vessel to tissue is exudation of leukocytes. Okay, that is the definition. Now the detail. These changes means the exudation of leukocytes also occur in some steps. So in that the first step is changes in the formed elements of blood. So here it is related with the hemodynamic changes of vascular events. What happened in that? If you remember, in vascular events, there is a vasodilatation, and due to vasodilatation, there is increased blood flow and increased blood volume, which is responsible for redness and the warmth. And due to that, there is a pressure increased, which increases the movement of fluid into the interstitial tissue, and then the flow becomes slow. So what happens with cells when these changes occur? What happens with cells, WBCs, when there is a dilatation, when there is a movement of fluid, when there is a slowing of blood flow? You can see here, right? So in early stage there is a slowing of blood stream, and due to that changes in normal axial flow of blood in the microcirculation takes place. So this step shows directly the fourth step of hemodynamic changes, that is the slowing of blood flow, right? Now before the slowing of blood flow, what happens with normal flow? So first of all, you should know about the normal flow. When the normally blood flows into the blood vessels, what happens with cells? Okay, so normally the normal axial flow consists of central stream of cells comprised of leukocytes and RBCs and peripheral cell-free layer of plasma close to the vessel wall. Means you can see the first picture. See what happens whenever the blood flows normally in the blood vessels. જ્યારે બી આપણા blood vessels માં blood flow થતું હોય તો cells અને plasma ની શું condition હોય છે blood vessels માં, that is explained in this first image. You can see the cells are flowed in a center position. Exactly, they are flowed in a central part. So there is a central flow of cells, right? And that is covered with the plasma. Means peripherally અથવા તો કહી શકો છો કે nearby the blood vessel wall, the close to the blood vessel wall, there is a cell free layer of plasma. Means there is only plasma present near the blood vessel wall, and the cells always flow in the center. So that is normally cells are in center, plasma in periphery. Okay, now what happens due to slowing of blood flow? When the blood flow becomes slow, the central stream of cells widens. કોઈ બી blood flow જ્યારે slow થશે તો એ જે center માં blood cells જે flow થાય છે that widens, that also goes near to the blood vessel wall, right? And peripheral plasma zone becomes narrower, and that is because of the loss of plasma by exudation, and this phenomenon is known as margination. Margination, you can see the second image of margination. See what happens in that. Compare the image with first one. First one, there is a central flow of cells. Now what happens in second image? See, the cells widens. So there is a central stream of cell widens, and due to that, and also the plasma starts to enter into the interstitial tissue. Plasma escape out. So when plasma escape out, plasma ઓછું થઈ જશે, then the cells widen and comes completely close to the blood vessel wall, and that is known as margination. And as a result of this redistribution, neutrophils of the central column come close to the vessel wall, and this is known as pavimenting. See, you can see the third image, pavimenting. Now what happens in pavimenting? That margination, all the cells come near close to the blood vessel wall, but in that particularly neutrophils come close to the blood vessel wall, and that is called as margination. You can see I have drawn these cells as N. Neutrophils are red colored with N, so that you can see that in image the neutrophils come closer to the blood vessel wall, and that is called as pavimenting. So in case of first step, changes in the formed elements of blood, what happens? First, you should write about the normal thing. In normal blood flow, there is a central stream of cells and peripheral cell-free plasma layer near or close to the blood vessel wall. Second, in that second step, when there is a slowing of blood flow, the central stream of the cells widens, and that's why they come close to the blood vessel wall, and also the plasma layer is reduced due to the loss of plasma into the interstitial tissue, and this is called as margination. And due to this margination, the more and more number of neutrophils come close to the blood vessel wall, and that process is called as pavimenting. Okay, so you should include 3 points in the first step, that normal flow, margination, pavimenting. Now the second step, rolling and adhesion. The rolling and adhesion, the name itself suggests rolling. So marginated and pavimented neutrophils slowly roll over the endothelial cells lining of the blood vessel wall. So as we have discussed in first step, that neutrophils come close to the blood vessel wall, and blood vessel wall is made up of endothelial cells. You can see there is a one layer of endothelial cells. So when the neutrophils come nearer to the endothelial cells, first they roll over onto the endothelial cells. Endothelial cells ઉપર આખા neutrophils roll થશે. એના ઉપર round round એ move થશે. That is the rolling phase. And this is followed by the transient bond between the leukocytes and endothelial cells, which become firm, and that is adhesion phase. So during that rolling, at one time there is a bond formed between the leukocyte, or you can say neutrophil, and the endothelial cells. And when there is a bond produced between these two cells, the neutrophils stick over there, right? And or you can say adhere over there, and that's why it is adhesion phase. So in the second phase, the neutrophils which comes nearer to the blood vessel wall roll over on the endothelial cells, and at one time there is a bond formed between the endothelial cell and neutrophils, which becomes then formed, and this is called as adhesion phase. So rolling phase, adhesion phase. Now there is a question, why this rolling and adhesion occurs? Why the neutrophils produce the bond between this bond with the endothelial cells? So the reason is the some factors which are secreted during this process, and the factors are selectins. Selectins are the substances mediated. Selectins are three types. Okay, so selectins are the substances which are of three types: P-selectin, E-selectin, and L-selectin. P-selectin is a preformed substance which are stored in endothelial cells and platelets. Means they are already synthesized and they are stored in endothelial cells and platelets. And when this process occurs, they are secreted from these cells. E-selectin is the substance which is synthesized by cytokine activated endothelial cells. That means cytokines are nothing but the inflammatory mediators. We will discuss that later on. But whenever there is a process of inflammation is initiated, cytokines are released. And when cytokines released, they activate endothelial cells and secretes E-selectin. So E-selectin are the substances secreted after activation from endothelial cells, and L-selectin are expressed on the surface of lymphocytes and neutrophils. So these three substances, P, E and L-selectins, are responsible for the rolling of neutrophils over the endothelial cells. So that rolling of neutrophils on endothelial cell occur due to these selectins. And like that, the another substances are integrins. And integrins are responsible for the adhesion phase. Integrins present on the endothelial cell surface. They are present on cell surface, and the receptors for the integrins are present on the surface of leukocytes. So you all know that whenever any substance come close to their receptors, specific receptors, then obviously they bind with their receptors. So integrin present on the endothelial cell and the receptors present on the leukocytes. So when it come close to the neutrophils, then the receptors and integrins bind with each other. So that neutrophil binds to that endothelial cell, and that is nothing but adhesion phase, right? And along with these substances, there is one another substance which is known as immunoglobulin superfamily adhesion molecule, such as ICAM1 and 2, which helps in localizing the leukocytes to the site of injury and thus helps in transmigration of these neutrophils. That means that the leukocytes particularly attracted towards the site of injury by these substances. So rolling adhesion occurs due to this substance. Now the third phase is immigration. Immigration is the after sticking of neutrophils to endothelium, the former move along the endothelial surface. You can see in the picture that blue colored are the neutrophils. So that neutrophils first stick to the endothelium, and then move with the endothelial surface. And then after finding the suitable site or suitable space, neutrophils lodged between the endothelial cells and basement membrane, and then crosses the membrane by damaging it locally with secreted collagenases and escape into the extravascular space, and this process is called as immigration. So you can understand the endothelial, first of all there is the adhesion phase, means the neutrophils bind to the endothelial cell, right? Now neutrophils find a space. We have already discussed in vascular permeability, there is a change in vascular permeability. There is a gap produced between the two endothelial cells. So when neutrophils come binds with the endothelial cell, it finds the space nearby that endothelial cell, and from that space it is escaped out. એ ત્યાંથી બહાર જતો રહેશે, right, from that space. So that WBCs from blood vessels transferred into the interstitial tissue from that space, and that process is called as immigration. And during that transfer, if the basement membrane is intact, if there is no space available, then what happened? WBCs secrete the enzymes collagenases, and that collagenases damage the basement membrane and create the space, and from that space it escape out. So likewise, ultimately, whenever the WBCs or you can say neutrophils bind with the endothelial cells, it crosses from the blood that blood vessel wall into the interstitial tissue, and that process is called as immigration, right? And simultaneously to immigration of leukocytes, escape of the red blood cells through the gaps between endothelial cells occur, which is known as diapedesis. That means sometimes if there is a increase or very high change in vascular permeability, then what happens? Along with WBCs, RBCs also escape out into the into the interstitial tissue. And if RBCs escape also along with WBC, then that process is called as diapedesis. So if only WBCs escape, it is immigration. If WBCs and RBCs both escape into the interstitial tissue, it is called as diapedesis, right? It is a passive phenomenon in which RBCs being forced out either by raised hydrostatic pressure or through the endothelial defect left after the immigration. And diapedesis gives the hemorrhagic appearance to the inflammatory exudate. That means that whenever there is inflammation, the fluid is accumulated, that fluid looks red due to the presence of RBCs. So when there is a diapedesis process occur, inflammation looks very red due to the presence of RBCs, and if only WBCs escape, then there is a presence of only plasma like creamish fluid. Okay, so when there is a diapedesis, you can clearly see the red colored fluid accumulated in the inflammation. And the fourth step is chemotaxis. Chemotaxis is the transmigration of leukocytes after crossing the several barriers. Which several barriers? First endothelium, that is endothelial cells, second basement membrane, and also matrix, to the interstitial space mediated by chemotactic factors is called as chemotaxis. That means the process of immigration, matlab ke je WBCs from one side transferred to the other side, if that is immigration, if that immigration occurs due to some substance, if there are some substance present which attract WBCs to transfer this basement membrane and endothelial cells, or you can say blood vessel wall, then that process is called as chemotaxis. And those substances which are responsible for this chemotaxis are called as chemotactic factors. So the agents that acts as a potent chemotactic substance for different leukocytes are called as chemokines, and they are You can see the list here. Leukotrienes, and particularly leukotriene B4, platelet activating factor B4, cytokines particularly interleukin 1, interleukin 5, interleukin 6, soluble bacterial products, and monocyte chemoattractant protein, that is MCP1. These substances are chemotactic substances or chemotactic factors which are called as chemokines, and when these substances are present at the site of injury, they attract WBCs from the blood vessels into the interstitial tissue. So that WBC is transferred by attraction because of these substances, and that is called as chemotaxis. Right. So in case of cellular events, there are two processes occur. First, exudation of leukocytes, and second, phagocytosis. Exudation of leukocytes occur in 4 steps. First, the change in formed blood elements. In that we have seen 3 steps, 3 points: normal blood flow, then margination and pavimenting. Then second phase is the rolling phase and adhesion phase. Rolling phase is due to selectins and adhesion phase is due to integrins. The third phase is immigration, that is transfer of WBCs from blood vessel into the interstitial tissue. And the fourth is the chemotaxis, when this immigration occurs because of the presence of some particular substances which are called as chemotactic factors, and the process is called as chemotaxis with the examples. Clear? So that is about the exudation of leukocytes. Right? Now the second step, phagocytosis. Phagocytosis is defined as the process of engulfment of solid particulate material by the cells, and that is called as cell eating. That is also you have already studied in Sem 1 anatomy. There are two types of endocytosis process, pinocytosis and phagocytosis. Pinocytosis is the cell drinking, phagocytosis is cell eating. Right. So when there is engulfment of any solid substance, solid particulate material by the cells is phagocytosis. Right. Cells performing this process are known as phagocytes, and those cells which produce this effect is called as phagocytes. Right. Mainly two types of phagocytes present: polymorphonuclear neutrophils and macrophages. Neutrophils and macrophages, these are the two major phagocytes which produces the phagocytosis process. Phagocytosis process is the same for both types of phagocytes. Means there is no difference in the process if it occurs with neutrophils or if it occurs with macrophages. Okay, first of all, do you know the macrophages are the which cells? You have heard about neutrophils, the type of WBC. Macrophages are also the type of WBC, and that is monocytes. Monocytes, when present into the blood vessel, they are called as monocytes. But when monocytes leave the blood vessel and transfer into the interstitial tissue, they are then known as macrophages. So monocytes have two names. When they are present in blood vessel, they are monocytes. When they are present in macrophages, when they are present in interstitial tissue, they are macrophages. Okay. So the process of phagocytosis is similar for both these cells. Now phagocytosis occur in 3 steps. First step, recognition and attachment stage, which is known as opsonization. Recognition and attachment stage. Second, engulfment stage. And third, degradation or digestion stage. First, recognition and attachment stage. Leukocytes bind and ingest most microorganisms and dead cells via cell surface receptors. Means leukocytes that engulf any of this or they bind with any of that solid particulate by the receptors, their cell surface receptors. These receptors recognize either components of the microbes and dead cells or the host proteins called opsonins that coat the microbes and target them for phagocytosis. And this process is called as opsonization. So what happens here? Which substances are present on the microbes or the dead cells? The substances which must be phagocytized. જે substance ને આપણે અથવા તો જે microbes ને આપણે phagocyte કરવાના છે એને ગળી જવાના છે એ substance ના surface પર શું present હોય તો કે ત્રણ વસ્તુ present હોઈ શકે. The components, particular કોઈ components હોય, પેલા microbes ના specific components હોય, અથવા તો ડેડ સેલ્સ પર એવા કોઈ સ્પેસિફિક કમ્પોનન્ટ્સ હોય અથવા તો પ્રોટીન્સ હોય. Which proteins? The proteins opsonins. Opsonins are the proteins which are synthesized in human body, in our body, host body. And then in host, these opsonins, whenever any microbes enter into the our body, then and any components are not present on their surface, then these opsonins cover these microbes, produces the coat on the microbes. એની ઉપર આખું એક covering બનાવશે. And that process is called as opsonization. એ covering બનાવવાની process ને કહેવાય opsonization. These opsonins are present in the blood, ready to coat the microbes, or they are produced in response to the microbes. એ બંને હોઈ શકે છે. Either they are already present in the blood, એ already blood માં બનેલા હોય, or whenever the microbes enter into the body, they are formed in presence of that microbes. Okay. Then these opsonins are the particular. There are examples. Antibodies of IgG class, immunoglobulin G class antibodies. Antibodies particularly which bind to the microbial surface antigens. Second, breakdown products of complement protein C3. complement system છે આપણી આખી immune system ની જેમાં number of proteins present હોય એને complement proteins કહેવાય. અને એમાંનું જે complement protein C3 છે એની breakdown products એ બી opsonin નું કામ કરે છે, covering કરવાનું કામ કરે છે, as well as collectins. Collectins are the plasma carbohydrate binding lectins which bind to the microbial cell wall sugar products. એ એવા substances છે કે જે microbes ના cell wall જે છે microbial cell wall એની sugar product છે જેને collectins કહેવાય. So they also act as a opsonins. તો opsonin એટલે particularly એક જ substance નથી હોતું. They can be immunoglobulin G antibodies, they can be complement protein C3 or their breakdown products, or they can be collectins. Right. Now, after the coating of opsonin on microbes, the receptors of the opsonin which are present on the leukocyte facilitate the rapid phagocytosis of coated microbes. જ્યારે એ opsonin એ covering form કરી દેશે એના પછી after that formation of that covering, the receptors for that opsonin is present on the surface of neutrophil or macrophage. So they can identify now this substance present, now we should bind with this substance with the receptors. So they bind with these substances with the receptors. See, જે substance ના આપણે example લીધા એના receptors બી છે. For the receptor, for FC receptor for IgG, FC receptor is present on neutrophil and macrophages, that is for immunoglobulin G. Second, complement receptor CR1 and CR3. If complement protein 1 is there, then CR1. If complement protein 3 is there, CR3. And that binds with the complement proteins. And C1 is for collectins. So if the opsonization occurs with collectins, it binds with the C1 receptor on the surface of neutrophils. So that is the first step. The first step is recognition and attachment stage. Recognition, that means identification. તો આપણા બોડીના મેક્રોફેજીસ ને ન્યુટ્રોફિલ્સ કેવી રીતે આઈડેન્ટિફાઈ કરશે કે આ સબસ્ટન્સ ને મારે ગળી જવાનું છે? So they identify based on the proteins opsonins which covers the surface of the microbes. That process is called as opsonization, and through the receptors present for the opsonins on the surface of leukocytes, they bind with that substance and start the and continue with the next process, right? So the second process is now engulfment stage. Engulfment stage, the opsonized particle binds to the surface of phagocytes for engulfment. એ એના જોડે bind થશે, engulfment start થશે. This is done by the formation of pseudopods around the particle and finally forming the phagocytic vacuole. Now you should remember the endocytosis process of your first semester here. If you remember, they start to form the cell surface of that forms the deep and deep layers and then form the vacuole, which are connected with each other, leaving the vacuole inside. You can see that in the first picture. In the first picture, you can see the A site. In the A picture, immunoglobulin G opsonin binds with the Fc receptor, right? Or Collectin binds with the C1 receptor, or complement protein binds with the CR3 receptor. So either of this, the substance or you can say microbe bind with the one receptor. આમાંથી ને કોઈ બી એક સાથે bind થશે. Then in B picture, you can see they start to form pseudopod. Pseudopod means deep and deep layer. And then at one time, both the points of both the ends of that surface binds with each other, forming the vacuole. You can see the C picture. There is a formation of phagocytic vacuole કે જે આખા microbe ને cover કરીને એને અંદર લઈ લેશે. That bacterium comes inside by forming the vacuole, and the plasma membrane encloses the vacuole and then it breaks from the cell surface. પછી એ બંધ થઈ જશે અને cell surface પરથી ને છૂટું પડી જશે. So that vacuole lies free in the cell cytoplasm, and based on that, that vacuole becomes free inside the cell, and that vacuole binds with the lysosomes. Lysosomes are the organelle present in the cell, right? So in the cell, that binds with them vacuole and forms the phagolysosome, or you can say phagosome, and that you can see in the picture D, last picture, phagolysosome. So in case of engulfment stage, they bind the microbe that bind with the receptor is totally taken in by the formation of pseudopod and then the phagocytic vacuole, and then that phagocytic vacuole attached with lysosome and produces phagolysosome, and that phagolysosome then enters into the third stage, degradation or digestion stage. Okay. Now the third one, the degradation digestion stage. In this stage, the role of phagocytes or scavenger cells are completed by killing and digesting the microbes. In this stage, what happens in that phagolysosome, that microbes are killed by the, or you can say digested by the scavenger cells, means macrophages and neutrophils. Microorganisms, after being killed by antibacterial substances, are degraded by hydrolytic enzymes, and however the mechanism fails to kill and degrade some bacteria like tubercle bacilli. Means કે microorganism that killed, એ મરી ગયા પછી બી એના substances, some lytic products બાકી રહેશે. So those lytic products are then degraded by the enzymes, hydrolytic enzymes present in the lysosome. And when this phagocytosis is not possible for some strong bacteria, then they can produce the infection. And the example is tubercle bacilli, જે આપણને TB કરે છે. જ્યારે TB નું ઇન્ફેક્શન થાય છે તો આ ઇન્ફ્લેમેશન થી એ TB ના ઇન્ફેક્શન ને રોકવાની ટ્રાય કરે છે આપણી બોડી, બટ એ TB ના જે બેક્ટેરિયા છે એટલા બધા સ્ટ્રોંગ હોય કે જેનાથી ને એ નથી સ્ટોપ થતું, તો એ સિવિયર ઇન્ફેક્શન પ્રોડ્યુસ કરશે in the form of ટ્યુબરક્યુલોસિસ કે જેનાથી આપણને ટીબી છે એના સિમ્પટમ્સ દેખાય. Okay, so that is the degradation stage which occurs in 3 steps. Degradation, or you can say killing, occurs by three mechanism: oxygen dependent bactericidal mechanism, oxygen independent bactericidal mechanism, and nitric oxide mechanism. The first, oxygen dependent bactericidal mechanism. So the name itself suggests, you can understand, in this mechanism oxygen is required because it is oxygen dependent. This mechanism involves killing by the production of reactive oxygen metabolites like superoxide radical, H2O2, hydroxyl radical, HOCl, HOI, HOBr, HO શું છે? That is hypo acids. HypoCl હોય, HOCl હોય તો hypochlorous acid. HOI છે તો hypoiodous acid. Br છે તો hypobromous acid. So they are the acids. Okay. So whenever these substances, oxygen reactive substances are present, that involves this mechanism. The phase of increased oxygen consumption, that is respiratory burst by activated phagocytic leukocyte, requires the presence of NADPH oxidase, which is present in the cell membrane of phagosome. Now what happens in this mechanism? The first most common requirement is NADPH oxidase enzyme, and that enzyme is present on the cell surface or the cell membrane of the phagosome કે જે આપણે બન્યું હતું complex vacuole અને lysosome નું એના surface પર હોય, right? And NADPH oxidase reduces oxygen to superoxide ion. What it causes? NADPH ને use કરીને એ oxygen ને convert કરશે superoxide ion, right? And NADPH is converted to NADP. Now this superoxide ion, that is O2 ion, is subsequently converted to H2O2, which has bactericidal properties. So that oxygen O2 radical, superoxide radical, then converted into H2O2. H2O2, that means hydrogen peroxide. So that hydrogen peroxide has a bactericidal action કે જે પછી બેક્ટેરિયાને કિલ કરી શકે છે. But a killing જે છે એ બી બે ટાઈપનું છે. That killing with H2O2 occurs by the presence of myeloperoxidase enzyme or in the absence of myeloperoxidase enzyme. So based on that, we also divide this mechanism in two things: MPO dependent killing and MPO independent killing. In case of MPO dependent killing, when the enzyme myeloperoxidase present, this H2O2 is converted into HOCl, HOBr, HOI, means hypohalous acids, and water. So these hypohalous acids having the strong antibacterial agent that kills the microbes within the phagolysosomes, right? So when myeloperoxidase present, H2O2 is converted into hypohalous acid. And in case of MPO independent killing, the macrophages lack the MPO, hence they carry out the bactericidal activity by producing OH ions, that is hydroxyl ions, and superoxide singlet ions. Oxygen ma single molecule, singlet oxygen from H2O2, and that is in presence of O2 radical, and that reaction is called as Haber-Weis reaction, or in the presence of ferrous ion Fe+2, so that reaction is called as Fenton reaction. So again I am repeating here that killing stage or degradation stage includes three mechanisms: oxygen dependent, oxygen independent, and nitric oxide. Oxygen dependent, what happens in that? NADPH oxidase converts oxygen into superoxide radical, and superoxide radical is converted into hydrogen peroxide. And that hydrogen peroxide has two mechanisms: MPO dependent MPO independent. MPO dependent mechanism, MPO means myeloperoxidase. So that myeloperoxidase converts H2O2 into hypohallous acid, and that hypohallous acid are strong antibacterial substance which can kill the microbes. While MPO independent mechanism involves the conversion of H2O2 into OH radical and superoxide singlet radical in the presence of O2 radical, and that reaction is called as Haber-Weis reaction, and in presence of ferrous ion is called as Fenton reaction. Now the second mechanism, oxygen independent bactericidal mechanism. Some agent released from the phagocytic cells like lysosomal hydrolases, permeability increasing factor, cationic proteins do not require oxygen for their bactericidal activity. So some substances which involved here are lysosomal hydrolases, permeability factors. Now what they do, the permeability increasing factor activates the phospholipase enzyme, and phospholipase enzyme, as the name suggests, phospholipase, which break the phospholipids, and phospholipids are present in the cell membrane. So when cell membrane phospholipids are dissolved, there is a pores present in the cell membrane which increases the permeability. So like that, a permeability increasing factors which do not require oxygen. Second is lysozyme cause degradation of oligosaccharides present in bacterial coat. The bacterial coat માં present છે lysozyme which kills the oligosaccharides, and that's why the bacterial coat is disabled, or you can say degraded, and that's why the bacterial cell get dead. And third is defensins. Defensins are peptides that kill microbes by creating holes in their membranes. So they are the substance which directly kill the microbes by creating holes. So permeability enhancing factors, lysozymes, defensins are the substances which do not require oxygen, and they directly produces microbial death and killing. And the third mechanism, nitric oxide mechanism. Nitric oxide mechanism involves the substance nitric oxide, and that is produced by endothelial cells as well as by activated macrophages. So that macrophages and endothelial cells secrete nitric oxide in response to inflammation, and these substances act similarly to the reactive oxygen species. reactive oxygen species superoxide radical, OH radical જે રીતે કામ કરે છે એ રીતે જ નાઇટ્રિક ઓક્સાઇડ ની મિકેનિઝમ છે કિલિંગ ની. In experimental animals, nitric oxide has been shown to have fungicidal and anti-parasitic activity. Whenever this nitric oxide is used in animals during experiments, it has shown two activities. It also produces the death of fungal cells and death of parasitic cells. But its role in bactericidal activity in humans is not yet clear. But this is true for animals, but still for humans, its activity is not clear. Right? The dead microorganisms are then degraded by the action of lysosomal acid hydrolases, in which the most important is elastase. You all know that as per our anatomy, lysosomes contain dozens of enzymes, hydrolytic enzymes. So when these microbes are killed, the dead microbial products are then digested by these lysosomal enzymes, and that particularly elastase, and then they are completely dissolved in the body. Okay, so these three steps are present in phagocytes. We'll again summarize in brief. Phagocytosis include three steps. First, recognition and attachment stage. Recognition by the process of opsonization, and through that opsonization there are receptors present on the neutrophils and macrophages, and through that they bind with the these cells. That is first stage. Second, engulfment stage. After that binding with the receptor, they engulf this bacterial product in the form of vacuole and then attach with the lysosome, produces phagolysosome. And in third stage, the killing stage, or you can say degradation stage, there are three mechanisms: oxygen dependent, oxygen independent, and nitric oxide. And after killing, the bacterial products are then digested by the use of lysosomal enzymes. Okay, so that is about the cellular events. First, exudation of leukocytes, and second, phagocytosis. I hope you have cleared all the two topics. If you have any doubts about that, you can feel free to contact. Thank you.